My name is Asila, and these are my group, Mohammed Manas and Shadin Benham, and Hiba is supposed to be here with us, but unfortunately she's now in Vietnam. Anyway, we're going to present for you today how to turn your eyes blue. If you have brown eyes, how to turn into blue, which is a mutation, through discussing various mutations, and through discussing the genes and locus and how gene correlations happen with each other. And Mohammed Manas is going to start introducing our topic. Mohammed, sorry.
Today I want to talk about, first of all, I want to talk about the structure of the eye and where is the pigment is in the eye. So we have, first of all, we have the pupils. Around the pupils, we have the iris. It's a muscular structure. This structure contains pigments. This pigment is called melanin. Inside these melanin, we have a cells called melanocytes. And in these melanocytes, we have the melanosome, which contains the melanin. The blue eyes and the brown eyes and each eye contain the same amount of numbers of them in the melanocytes. However, which varies is the number of melanosomes and the amount of melanin inside the melanosomes. Eyes. We have two, they divided the eyes into two groups. One, which is colored. We have the brown, the hazel, and the green, and other colors. And the other eyes, which is uncolored eye, which is the blue. The colored eye contains 3 layers, and the uncolored eye, blue, basically both of them contains 2 layers, but in the colored eye we have a 3rd layer which contains the pigments. Once upon a time, زمان يعني, when we studied genetics in high school, we thought that the eyes is determined by only one single gene, and it's only Mendelian, because they thought that the brown is dominant and the blue is recessive. And when they meet in a heterozygous form, they produce brown and a heterozygous which contains the other colors and the blue color. But today, and this is basically incorrect, today they discovered at least that there is at least 8 genes that contribute in the formation of the eye color. One of those genes is the OCA2 gene, which the 3/4 that controls the eye from other genes. This OCA2 gene will produce a P protein. This P protein will produce the melanin, which will form the eye colors. Today, these genes, they found that there is a correlation, and we're going to find that through this research, that if there is a correlation between this gene and the HCER2 gene. And the mutation that happened previous ago, before 10,000 years, they found that there is a mutation happened here, which stops partially, not totally, the OCA2 gene, which will produce the blue eyes. If it stops completely, it will produce the albinism. and that's it. And my colleague Shaden will talk about the materials and the methods that will contribute in this research to find that if there is in the chromosome 15, if there is a relation between this gene and the HCR2 gene. Thank you.
Welcome.
Hello everybody. Now I'm going to give you a general idea about the methods that were used in the experiments we used in our research. In our research is based on 3 experiments actually, because we used 3 papers. Now I'm going to explain one of the experiments. It's the main one, and it includes almost the whole materials and methods that were used. The organisms were used in the study are mostly human organisms, which consist of 3-generation Danish family that was selected according to their eye color. The individuals were selected. The study selected the individual which has only brown and blue eye colors, and they excluded the other eye colors. The study provided many figures, and there is one main figure that shows the phenotypes of the eye color. Next, please. And this is the main figure. It consists of 4 subfigures. Figure A shows us the pure blue color. It's known as the complete lack of brown pigmentation. The figure B shows us the intermediate phenotype. That shows the blue color with a little small brown dots inside the blue color. Figure C shows us also another intermediate phenotype, which is like mix of blue and brown eye color. The fourth one is the brown homozygous phenotype, which, as you see, is complete brown pigmentation. Next. Okay, these previous 4 phenotypes were selected for the identification of the nucleotide positions. This process or technique is called the DNA sequencing. After doing the DNA sequencing process, the sequenced regions represent the exons, the coding exons, which can be after that synthesized to normal proteins, and it also includes some conserved areas located in the entrance of our 2 main genes. Our 2 main genes are the OCA2 and the HERC2 genes. Now the PCR process. The PCR process is the polymerase chain reaction. It was carried out in the experiment under specific standard conditions, and it's a scientific technique to amplify or to magnitude the size of the DNA sequence. It was used and it was applied on specific area of the DNA molecule in order to see the figures or the sequencing in more clear view. Now one of the main things in the DNA sequencing or in the PCR is the promoter activity in the cell culture. The promoter, you all know the promoter is the space where we start, where it can be recognized where we start the translation process. So human OCA2 promoter region was PCR amplified using the genomic DNA from a brown eye homozygous individual. So we used a homozygous brown individuals, which you already see in figure 1. It's the 4th picture, which shows the complete presence of brown pigmentation. The PCR product was subcloned and confirmed by the sequencing, and the OCA2 promoter fragments were further subcloned. So we used the DNA sequencing first, then the PCR to amplify them, and they were subcloned after that. We have the 675 or 675 base pairs PCR fragments. They were cloned and sequenced before subcloning using the PAMHI restriction site upstream of the OCA2 promoter. After doing all these processes and techniques, we had some specific results that Asil will introduce to you. Thank you.
Hello, I'm going to discuss with you the overall process, how to correlate between and what is actually the correlation between the OCA2 and the HERCO2. They found that there are some regulatory elements within the HERCO2 gene which inhibits the activation of the OCA2 expression, which actually is going to produce the blue eye color. Scientists have mapped the genome for more than, first they started genoming the complete genome for 5 Danish families, and then they expanded it to other 40 Danish families. And they found that chromosome 15q is a chromosome that carries the 2 genes which are the OCA2 and the HERCO2. Here I said that they use results and scores from the LOD. The LOD is a statistical genetical method to find, if you have a chromosome, there's a specific locus in it, you can find which are 2 genes beside each other and how they affect each other. And they use this technique to find the candidate genes for the blue eye color mutation, and they discovered that it's 15q. The scientific association method, we've talked about, they found some regulatory elements. The regulatory elements are these 2 regulatory elements. They are strong regulatory elements, and they are the most preferable candidates for the blue eye mutation. Next. Now, we've talked about the mutations. Where did it start? How did it start? And for sure it's an evolution because it's taken for our day. This mutation, blue eye mutation, started in the northwest of the Black Sea region. It started there because it's a great agricultural migration to the northern part of Europe. Took place in the regions about 6,000 to 10,000 years ago, which caused the evolution of the blue eye mutation. Now, why was the evolutionary thing? Because many theories actually, natural selection didn't help a lot, but they've explained, some theories they've explained that the main results, the main causes behind this evolutionary selection was the UV exposure to radiations, which actually causes skin cancer, vitamin B deficiency, and sexual selection have been mentioned. Other things that for each gene we know that there is a promoter. Now, what is the promoter for this gene that causes the blue eye color mutation? They found that BEH2 might be the promoter region for the OCA2, which is the blue eye allele that lowers the amount of OCA2 expressed in the iris. Each time you lower the OCA2, you increase the blue eye colors and decrease the brown eye color. Now, they said that TGA allele of the BEH2 are the most common, and the BEH are the most common signal for the selection in Europe and Southwest, and a signal been seen at the BEH and the BEH3 is not as strong as TG and the BEH, which actually cause the blue eye color. That's why you can see people with blue eye colors more in Europe rather than here. The TG allele of the BEH2 contains a strong LD with the blue eye casual mutations. Now here's one of the experiments that actually show the SPIN, and it's an abbreviation for single polymorphism nucleotide, which actually is the main cause for one of the causes for the blue eye color. As you can see, the first 2 are the Homo sapiens, which are humans, and here they've sequenced their genome for the DNA sequence to find the colors. They found that people with the blue eye color, they have this nucleotide mutation. As you can see here, it's TAAG rather than A, and then TG, which actually causes the blue eye color, this G. Other people who carries the brown eye color, they carry the TAAA-TG. So blue eye colors, they have spins for the in their DNA for a G rather than an A. And as you can see, other organisms, they have A, not a G. That's why they have the brown eye colors. As a conclusion, I just want to say that they've proved through finding the DNA sequence and genome mapping, they found that gene HERC2 It is the gene that inhibits the OCA2 gene, and when it inhibits it, it reduces what it doesn't. It inactivates it and produces more blue eye colors. If OCA2 was not activated, your eyes will stay brown. It will show brown eyes rather than a mutation, and also through this mutation causes the blue eye color in the northeastern sea. Thank you all, and Mohammed Nasra will continue.
The same gene,
OCA2, it is the one that is if it is inhibited, it causes the blue eye color mutation.
so if we inhibit it,
Yeah, if you express more and inhibit it, yeah, it does. And actually, this is the main thing. If you could change G, change the A into a G, adenine to guanine, yeah, this would definitely show blue eye color because you inhibit.
I can choose my children's eye color.
If it's ethical, yeah.
There's been all the risk of dangerous mutations. You don't know what will happen.
Any other questions? Questions? Other questions? Muhammad.
So as Asid said, that we have the HCA2 gene, which will inhibit partially the OCA2 gene, which will produce the blue eye. However, if it does not inhibit it partially, it will produce the brown eyes. But if this inhibition was completely, it will produce the albinism, with the white people and their hair is white and they have a red eye, but this is not a red eye. They show that the light is reflecting inside the eye and there is no pigment, so what will produce is will show the inside blood vessels, which is the retina. However, for this, in the limitation of this study, they did not talk about how partially will affect. They did not talk about other colors and how partially this mutation will affect. If it partially, how this partially will, the range of this partially. And another thing, they talk about the Danish and Jordan and Turkey, but they, what if they take a range from the quarters? Because if you go to the quarter, the more sunlight will goes, and that will affect the eye color. So it will, the eye color will be determined by the environmental factor here, as it shown in the quarter. And that's it. Thank you.